not so many lah. Okay, so I will explain you the basic. Lepas tu nanti further information saya akan attach few papers lah. Okay, alright. But because hari ni kita hopefully kita akan habis topik 3 topics. Okay, so I have to be ni lah topik ni macam penting dan banyak akan masuk exam lah. So I hope that you can pay attention lah. Okay, alright. So kita ni lah start dengan Zulharis. Boleh baca doa dulu? Zularis, ada ke? Mas Rizal?
Bismillahirrahmanirrahim. Alhamdulillahirrahmanirrahim. Hamdan syakirin. Hamdan wafil makafi umazida ya rabbana lakal hamdu kama yanbaghi li jalali wajhika wa azimi sultanik. Allahumma aftah alaina hikmataka wa syu'a alaina min huzaini rahmatika ya arhamar rahimin. Rabbi shrah lana sudurana wa yassir lana umurana wahlul 'uqdatam min alsinatina yafqahu qawlana. Allahumma fa'alna bima 'allamtana wa 'allimna ma yanfa'una wa zidna ilman nafi'an wa zidna fahman wasi'an bi rahmatika ya arhamar rahimin. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Sallallahu alaihi wasallam Muhammad wa ala alihi wasallam walhamdulillahirabbil alamin.
Thank you. Okay, right. So hari ni we going to continue this morning. We going to continue topics yang kita discuss semalam on tools for plant genetics. Okay, like I said that there are many tools actually, but I will introduce some of the common one lah yang dan yang basic lah. Okay, alright. Okay, let me share my slide. Okay, boleh nampak ke slide saya?
Boleh.
Okay. Alright, so the first tool that I introduced was yesterday, okay, was PCR. Okay, I hope you able to understand the principle of PCR. Okay, so the components, the profiles, okay, what happened for each cycle, okay, and then you should be able to design. Let's admit someone. Okay, you should be able to design primers. Okay, you should be able to calculate aniline temperature. Okay, and then you should be able to macam mana nak calculate amount of stock. Okay, to prepare PCR reaction. Okay. Right, if kalau ada yang tak faham, let me know, okay? Alright. Okay, so right, and also after that, okay, we also discuss about how to do reverse transcription. Okay, so for reverse transcription, it involves 2 steps. Okay, first one is to isolate the mRNA. Okay, so how we isolate mRNA is based on the features of mRNA. One of the feature is actually in eukaryotic cells, the mRNA contain poly A tail, right? So based on this feature, okay, we'll be able to isolate mRNA from total RNA, okay? Okay, using oligo-DT base, all right? Okay, and then after that, if you successfully isolate mRNA, okay, the next step is to synthesize the cDNA. Okay, so these are the steps which we already discussed yesterday. Okay, so basically, in the reaction, okay, you add oligo-DT primer. Okay, so the primer will bind to the poly-A tail of the mRNA. Okay, and you add reverse transcriptase. So reverse transcriptase will add the nucleotides based on the complementary with the mRNA. Okay, to synthesize the one strand of DNA. Okay, so then to remove the mRNA, okay, now we don't need the mRNA. Okay, we remove by alkaline treatment. Okay, so the now leaving only one strand of DNA. Okay, DNA is actually, this DNA is actually complementary DNA, cDNA. Okay, so one single strand of DNA is actually not stable. Okay, it will form hair loop, hairpin. Alright, you nampak dia akan berpusing macam tu, hairpin formation. Okay, so using the first strand of DNA yang tadi tu kita synthesize, DNA polymerase will use as a template to add nucleotide. Okay, so now you have the two strands of actually you dapat dua DNA, but you want to remove this hairpin. Okay, so you treat and then fill the end with polymerase, you dapat double stranded cDNA. Okay, any question? Tak ada, clear? Okay?
Yeah.
All right. Okay, so I think that's very simple. So, but the disadvantages of using that method yang tadi tu, this figure, is actually it requires hairpin formation to prime second strand synthesis. Okay, and the need to cleave the hairpin. Okay, so we need to remove the hairpin with S1 nuclease, which can cause loss of sequence at the 5' end of the mRNA. Okay, so another strategy to avoid this problem, which you can lose some of the sequence at the 5' end, is to use homopolymer tailing, which is to add a tail to the 3' end of this first cDNA strand. Okay, so if you add, if the aminotransferase is provided with DCTP, for example, with a string of C residues to the 3' end. Okay, so this enable oligo-DG primer to initiate second strand synthesis. Okay, so this is what we do. Okay, another method, okay, for cDNA synthesis compared to this one is to use this one. Okay, so you sama juga step, you ada oligo-DT primer, they produce reverse transcription. Okay, removal of mRNA. Okay, ni kita remove mRNA, so you add homopolymer tailing. Okay, so where you add to the prime hujung ni adalah C. Okay, DCTP. Okay, so then using oligo-DG primer, okay, which is G akan base pair dengan C, okay, dia akan able to synthesize another strand of DNA. So this is cDNA synthesis using homopolymer tailing. Okay, this is another way how to synthesize cDNA. Okay, all right. Or you can degrade the RNA strand. Okay, by using RNase H, we leave small RNA fragment. Okay, this they akan cleave, they akan degrade RNA strand, but akan leave some of the RNA fragment as the to act as primers. Okay, for second strand synthesis, which you can also do where you degrade this mRNA. Okay, dia akan hasilkan fragments. This fragment akan bertindak sebagai the primer untuk synthesize another strand of DNA. Okay, alright. So that is what we call as reverse transcription. Okay, so but the problem with reverse transcription using the mRNA preparation, it can the RNA can be contaminated with genomic DNA also. Okay, so the PCR can also result in amplification of the contaminating DNA. Okay, so when working with eukaryotic material, this can be overcome by designing the primers, okay, so that the amplicon can expand the intron, because you know mRNA tak ada intron so only genomic DNA ada intron. Sebab mRNA adalah dia dah diproses kan. Okay, so amplification of any genomic DNA will be prevented. Okay. Alright, so that is reverse transcription. Okay, so now I'm going to introduce you to another technique, which is gel electrophoresis. Okay, so you run electrophoresis previously when you isolate the genomic DNA, right? Okay, and I assume you also run electrophoresis with Dr. Tamil, betul ke? Hari tu you isolate plasmid ke? You isolate apa? Ingat tak yang you buat lab dengan Dr. Tamil petang Jumaat tu apa? Untuk herbs and natural products.
Tapi tu tak guna electrophoresis, letak je atas terus nanodrop tu.
Oh, you tak check ada ke tak, band ke tak ada?
Tak.
Oh, okay. Tapi you run electrophoresis with me, right? So remember yang saya look jauh tu, okay, so that is electrophoresis. Okay, so the word electrophoresis is actually consists of two words. Okay, the Greek word of phorus, electro, you know lah, electricity, and phoresis is actually from the Greek word phoros, okay, which is to carry over. Okay, so electricity will carry over either nucleic acid, it can be DNA, RNA, or protein. Okay, so we use electrophoresis is actually to separate charged molecules lah. Okay, mainly nucleic acid and also protein. Okay, so this technique of electrophoresis is being used to study properties of single charge molecule, or actually you want to separate molecules lah. Okay, for us macam hari tu kita nak tahu kita isolate tak successfully isolate genomic DNA ke tak. So we run electrophoresis, okay, gel electrophoresis. Okay, you know this is just repetition. Okay, nucleic acid, we know that the basic unit is nucleotide. Okay, there are two types of nucleic acid, DNA and RNA. Okay, so because of the presence of the phosphate group at the nucleic acid, okay, so the large negative charge. Okay, so nucleic acid, DNA and RNA is negatively charged. So it will migrate towards the positive pole. Okay, while for protein, kita tahu the basic unit is amino acid. Okay, and protein contain both acidic and also basic residues. This is what we call as amphoteric compounds lah. Okay, so each protein has its own characteristic charge properties depending on the number and type of amino acid lah. Okay, kita boleh tahu dia punya pH berapa semua tu. Okay, so untuk DNA molecules, normally kita run gel electrophoresis, what we call as agarose and acrylamide. Okay, so when you want to conduct electrophoresis, okay, these are the equipment yang kita perlukan lah. Okay, so you seen previously the electrophoresis chamber. Okay, we need power supply, power pack. Okay, and we need the tray where we cast our gel. Kita buat kita punya gel lah. Okay, and to do the wells, okay, we need what we call as sample combs. Okay, and required buffer. Okay, and then we need loading die. Okay, loading die yang warna biru tu, normally kita pakai biru. Okay, which contain actually in the loading die, it's actually ada contain normally glycerol. Okay, so that the sample, okay, for example, if you want to check genomic DNA, your genomic DNA will be able to fall lah macam pemberat dance, okay, to the sample wells and tracking dyes lah, which is allow monitoring lah. It can be either blue yang kita ada kat lab warna biru ataupun can be orange, normally the colored lah. Okay, and then we have to add ethidium bromide. Okay lah, I see ethidium bromide ni is actually carcinogenic lah. Okay, so some lab sekarang dia tak guna ethidium bromide lah, sebab bila dia use ethidium bromide, semua equipment akan contaminated with ethidium bromide. Okay, so ethidium bromide yang kita add, because ethidium bromide ni will be able for you to view the chart. Okay, so the function of ethidium bromide ni actually dia akan intercalate between DNA. Okay, so bila dia intercalate between DNA, okay, bila you view bawah alpha imager, dia akan fluoresce. Okay, kalau you be able to extract DNA ke, you nak check PCR product ke, that's why kita add ethidium bromide lah. And then we need a transilluminator, which is in our lab we use alpha imager lah, okay, untuk to view the chart. Okay, any questions so far? Any question? Tak ada? Clear? Alright. Okay. Okay, saya move to the next slide. Okay, so basically the migration of DNA, when you run the DNA sample, okay, through agarose gel, it will depend on 4 main parameters. It depends on the molecular size, agarose concentration, conformation, okay, the shape of the DNA, and also applied voltage. Okay, so I will explain one by one. Okay, so depending on molecular size, it's actually molecules of linear duplex DNA travel through agarose gel at a rate which is inversely proportional. Okay, so kalau dia punya size lagi besar, molecular weight dia lagi besar, dia akan move slower. Okay. Alright, so maksudnya kalau you ada sample, ada banyak DNA sample, okay, so DNA lepas tu bila you run gel, yang DNA sample yang ada size bigger, okay, for example you nak run gel 1, you run gel, you expect you akan dapat DNA with the size of 1000 base pair, okay, and gel number 2 you run DNA yang you expect actually the size will be 500 base pair. Okay, so bila you run gel, band yang 500 base pair tu, well number 2 akan ke bawah, yang 1000 base pair ke atas. Okay, because it depends on the molecular weight. Okay, any question? Tak ada? Clear? So bigger size of DNA will move slower compared to the inilah, alright, smaller size. Okay, so in terms of concentration, kalau kita tahu kalau the agarose concentration is higher, okay, katakanlah you prepare 2 gel, okay, satu gel is actually 100% agarose, the second gel is 2% agarose. Okay, and you run DNA sample yang similar, you run DNA yang ada size 1000 base pair. Okay, you run for 1 hour with the same voltage. Okay, after 1 hour, gel number 1 tu kita akan nampak dia akan dah move ke bawah compared to yang 2% tadi. Okay, sebab kalau lagi keras gel tu, dia punya movement of DNA akan slower. Okay, so different concentration resolve different sizes of fragment. Okay, so higher concentration will facilitate separation of small DNA. Okay, kalau you tahu you punya DNA tu size besar, janganlah pakai yang jangan pakai agarose, jangan prepare concentration of agarose yang tinggi, sebab dia akan move lambat. Okay, so low agarose concentration allows resolution of larger DNA. Okay, so kalau you want to run larger than size of DNA, you prepare 1%, for example. Okay, but if you want to separate small DNA, normally kita use higher concentration lah. Okay, so this is imagine ada gel number 1, number 2, number 3, and number 4. Okay, so the gel is actually with different concentration. Now, gel number 1, 2%, 2, 1.5%, 1%, and 0.5. You are running the same sample. Okay, for 1 hour with the same voltage, so you can see that with 0.5% after 1 hour, band tu akan ke bawah. Okay, but for 1% akan sikit, 1.5 and 2%. Okay, any question? Tak ada. Clear? Okay. Alright, and then conformation. Okay, so there are three types of conformation of DNA. DNA can be closed circular, can be neat circular, and can be linear DNA. Okay, so this is linear molecules, linear, okay, neat, okay, and then you have supercoiled plasmid. So this different form of same DNA will migrate at different rate. Okay, so linear form yang linear akan migrate at the slowest. You know kenapa? Because dia punya surface area lagi banyak. Okay, while supercoil will migrate the fastest. So bila you run gel, okay, so this you akan nampak macam ni lah. Alright, so the migration of DNA, kalau dia ada tiga form, okay, supercoil akan bergerak paling bawah, ni linear macam ni. Okay, with the same size but different conformation. Okay, clear? And then applied voltage. Okay, kalau you nak cepat, okay, you run higher voltage. Okay, for 1 hour you set 120 volt. Okay, ataupun kalau you ada masa, okay, tak apalah, you run 100 volt, for example. So typically kita set the voltage according to the size of the agarose. Or the length of the gel, which is 5 volts per cm. Okay, so as voltage increase, larger fragments will migrate proportionally faster than small fragments. Okay, for fragments larger than 2 kb, the best resolution can be achieved by applying no more than 5 volts per cm. Okay, so cm is actually the value between the two electrodes. Okay. Alright, so that is for agarose untuk run nucleic acid. Any questions so far? Tak ada?
Doktor,
Okay, ya.
tadi yang doktor kata dekat DNA conformation tu yang slowest tu sebab dia ada total surface area banyak
Yelah, pasal bila dia linear, dia surface area dia ni.
Okay.
Right. Okay, so any question? Any more? Tak ada? Okay, so we move for protein pula Okay, so for protein, it's actually, it utilizes polyacrylamide. Okay, dia different. Protein kita tak pakai agarose. Kita pakai the type of gel tu, kita pakai polyacrylamide. Okay, so they actually based on polymerization of acrylamide. Okay, and in the presence of small amount of cross-linker, what we call as bis-acrylamide. Okay, so protein gels are usually performed under denaturing condition. Okay, in the presence of SDS, sodium didosyl sulfate. Okay, so kita add SDS. Okay, in the presence of SDS, disulfide cleaving reagents will solubilize. It will denature and dissociate the most protein into single polypeptide chain. Okay, so this is the function of SDS. Okay, so to denature and also to dissociate most protein into single polypeptide chain. Okay. Alright, so majority, okay, if the amount of SDS yang kita add depends on the gram of protein. Normally kita add 1.4 gram of SDS to 1 gram of protein. So like I mentioned, the function of SDS ni, it will denature most protein, okay, so that it will give all protein the same shape, okay, and it will bind directly to the protein at the constant rate at approximately 1 SDS molecule per 2 amino acid residues. Okay, alright, and electrophoretic migration through gel is determined by the size of SDS protein complex. Okay. Alright, so SDS akan denature proteins, so the proteins that denatured by SDS are net negative, and since the binding of the detergent is proportional to the mass ratio of the protein, the charge to the mass ratio is constant. Okay, alright, so it's totally different. Okay, for staining of protein, kita use Comasi blue or silver stain. Okay, protein ni untuk prepare gel dia lagi kompleks lah daripada DNA. Okay. Alright, so you can use Comasi blue ataupun silver stain for staining, but silver staining is more sensitive. Okay, 2 times sensitive compared to Comasi blue stain. Okay. Alright, so this is comparison between agarose and polyacrylamide gel. Okay, so for agarose, agarose is actually a linear polysaccharide. Okay, normally kita dapat agarose ni daripada microalgae, seaweed. Okay, so agarose made up of basic lipid unit agarobiosis. Okay, while polyacrylamide consists of, macam kita cakap lah, polymerization between acrylamide and also bisacrylamide. Okay, and agarose is being used to separate very large molecule, for example nucleic acid, large protein and protein complexes. Okay, but polyacrylamide mostly being used to separate proteins lah, most proteins. Okay, and agarose has a large range of separation but low resolving power, while polyacrylamide has small range of separation but very high resolving power. Okay, in terms of concentration use, normally kita prepare agarose at the concentration of 0.5 to 2%. Untuk polyacrylamide pula different, normally kita prepare between higher concentration, which is between 3.5 to 20%. Yeah? Okay, so this is a comparison. Okay, normally kita run agarose gel in horizontal format using this, the gel chamber. Untuk acrylamide gels run vertically. Okay, that's why saya kata kalau kita prepare acrylamide gel lagi kompleks lah, kita akan prepare dia vertical. Okay, so acrylamide gel kita akan buat vertically lah. Okay, so this is acrylamide gel. Kita akan prepare 2 gel lah. Kita akan panggil stacking gel and also kita akan prepare running gel. Okay, so that is will be more complex. So first kita akan prepare running gel. Okay, lepas tu kita kena pour pula dia macam vertical kan. So lagi kompleks, dia memang susah. Okay, so kita akan prepare running gel dulu. Bila dia dah keras, baru kita pour stacking gel. Okay, so the pH of the stacking gel and running gel also different. Okay, because kita nak separate the protein kan. So the stacking gel is normally the pH is we prepare at 6.8, while running gel kita prepare at the pH of 8.8. Okay, so this is what we call as discontinuous system because kita ada 2 gel. Okay, so ni saya bagi je you formula. Okay, you don't have to remember because katalah tiba-tiba you buat project FYP buat involve protein. Okay, so resolving gel atau separating gel, okay, which is yang bawah tu, normally kita prepare concentration dia higher lah, 12.5%, okay, at the pH 8.8. Okay, so this is the formula lah, you don't have to remember. Okay, while the stacking gel, the concentration is lower and the pH also lower, that is 8.8, now 6.8. Okay, so these are the components of stacking gel yang atas tu. Okay, any question? Okay, that are being used to separate protein lah. Kalau you nak we extract protein, okay, you want to check whether it's successful or not. So you ni lah, okay, boleh tengok using SDS dish. Okay, any question? Tak ada?
No.
Alright, clear. Okay, so another way to separate protein, we can use a technique what we call isoelectric focusing. Okay. So this technique is actually where you separate proteins, okay, based on pH gradient formed by the ampholytes. Okay, so at a specific pH, what we call as isoelectric point (PI), protein actually has no net charge. Okay, so dia tak boleh bergerak, dia jadi immobile. Okay, so at pH below PI, it is positively charged. So the protein akan move towards anode. Okay, the pH above Pi, the protein will become negatively charged. So it will move towards cathode. Okay, so for isoelectric focusing point, they akan migrate to a pH region where they have no net charge. Okay, so this is what the technique of what we call as isoelectric focusing. Okay, and then we have 2D gel electrophoresis. Okay, where this technique is being used to separate complex mixture of protein. Okay, so it will allow separation of proteins which reside in similar location in one-dimensional gel, and these are separated in the two-dimensional gel. Okay, so the first dimension, tube gel is actually electro-focused. So in the first step, okay, the protein migrate in electrical field in a gradient gel. Okay, and then the protein will stop the migration when they reach the point in the gel that has a pH equal to their Pi tadi tu, isoelectric point. Okay, so this is a tube gel. Okay, so this is the first dimension. Okay, and the second dimension is what we call as SDS, a slab gel. Okay, so in the second step, the gradient tube gel yang tadi tu is carefully removed, okay, and is placed over a slab gel, and electrophoresis is repeated. Okay, so in this gel, each protein produce spot, okay, which can be visualized in the gel by staining, either using silver staining or Komatsu blue, or when the proteins are radioisotope labeled by autobiography. Okay, so this is the second dimension using slab gel. Okay, so at the end of the experiment, what you will get is the spots. Okay, so akan bergerak dekat spots. Okay, so you akan identify the protein. Okay, any question? Any questions so far?
No.
Okay, clear? Okay, so sekarang I will explain you about sequencing. Okay, so that you'll be able to do the video on sequencing. Okay? Alright, and also, okay, this is clue lah. I have a question dekat exam on sequencing. Okay, so I hope you should be able to understand what I explained. Ni lah, kalau you have any question, you should ask me. Okay, so sequencing ni, sekarang ni, you know, the sequencing is actually kita sequence. Okay, either we sequence the nucleic acid or we sequence the protein. Okay, so we want to know the sequence. For example, we want to know the ATGC and so on. Okay, or we want to sequence the protein. Okay, we want to know the amino acid. Okay, for example, proline, tyrosine, and so on. Okay, so sequencing is actually to characterize a molecule, whether to determine the order of amino acid in protein or to determine bases in nucleic acid. Okay, so why is it important to do sequencing? Okay, because based on the nucleotide sequence or based on the protein sequence, okay, we be able to know the function of a gene. Okay, so previously, masa zaman dulu, okay, the sequencing is actually to sequence, for example, an organism is very expensive. Okay, but sekarang ni ada banyak technology available. Okay, so the process of sequencing become more efficient and it's become cheaper lah. Okay, but the sequencing technique ni is actually was initially developed by Frederick Sanger. Okay, so that's why bila kita cakap sequencing, kita selalu relate dengan Sanger sequencing technique. Okay, because this technique is developed by Frederick Sanger. Okay. Alright, so these are the two main principles, okay, of sequencing, where you should remember. Okay, I mentioned yesterday kan about the process of replication. or process PCR, okay, where if you want to synthesize DNA, it requires a primer. Okay, so the primer will annul to a template strand. Okay, so the synthesis of new strand work by adding bases to the primer. Okay, remember kita ada template strand yang kita nak amplify. Okay, and then kita add primer. Okay, alright, so the primer akan annul to the template strand. Okay, and then polymerase akan start to add the nucleotide based on complementary of the template strand. Okay, so it will extend the sequence. Okay, and the second principle yang you should remember for sequencing, okay, the addition of bases to the growing strand occurs by formation of what we call as phosphodiester bond. Okay, between the 5' phosphate of the nucleotide to be added and the 3' hydroxyl group on the existing molecule. Okay, so the substrate yang being added is actually 5' DNTP, which is deoxynucleotide. Okay, with 3 phosphate at the 5' position of the deoxyribose. Okay, so you nampak this is a normal DNA synthesis. Okay, so katakanlah you ada template strand kat sini, imagine template strand. Okay, and the normal DNA synthesis, okay, akan being added. Okay, katakanlah you ada primer ni. Okay, so kat sini ada C, ada A, so T akan being added. Okay, sini the template ada G, okay, C akan being added. Okay, akan being added by the formation of phosphodiester bond, and the substrate being added is deoxynucleotide. Okay, it can be DGTP, it can be DCTP, it can be DTTP, or it can be DATP. Alright, clear? Okay, so but the concept of sequencing is based on the concept addition of dideoxynucleotide. Okay, so the difference between deoxynucleotide and dideoxynucleotide is actually the deoxynucleotide at the 3' ada oxygen. Okay, but dideoxy ada hydrogen saja. Okay, tak ada hydroxyl group. Okay, clear? Alright, so the sugar part of the natural substrate is 2' deoxyribose. It does not have hydroxyl group at the 2' position, but does have a 3' hydroxyl group. Okay, but if 2', 3' dideoxy is the substrate, the DDNTP can be incorporated into its 5' phosphate and the 3' OH of the previous residue. Okay, this reaction will produce a strand that does not have 3' hydroxyl at the 3'. Okay, so it will terminate the DNA synthesis. Okay, so you bayangkan. Okay, so this is the normal DNA synthesis. Okay, DNTP. Okay, akan ada 0 hydroxyl group. But if DDNTP, dideoxy-NTP, is being added to the growing chain, okay, it will block the DNA synthesis because dia tak ada 3' hydroxy for further addition of nucleotide. Okay, clear? So any question?
No.
Tak ada? Okay. Alright, so I'll show you a video so that you boleh faham better.
To sequence DNA by the chain termination method, the DNA must first be obtained in single-stranded.
Okay, so these are the DNA that you want to sequence, eh?
A mixture containing the single-stranded DNA, DNA polymerase, the 4-deoxyribonucleotides A, T, G, and C, and a single short primer is prepared. The primer has a nucleotide sequence that is complementary to the 3' end of the region to be copied, and is required so that DNA polymerase can initiate DNA replication. Equal amounts of this reaction mixture are placed in each of 4 tubes. And
so maknanya you ada 4 tube. Okay, so you ada 4 tube. Okay, semua ada contain semua tu. So you label satu tube A, G, C, A, and T. Okay, dalam semua tube ni ada komponen yang tadi tu. Okay.
Different dideoxyribonucleotide is added to each tube.
Okay, but tube yang you label sebagai D, you add DDGTP. Okay, so tube yang you label sebagai C, you add DDCTP on top of yang tadi komponen tadi. Okay, clear?
When a deoxyribonucleotide is inserted into the growing.
Okay, so this is for example in tube yang ada label sebagai C, you add DDCTP tadi. Okay, this will happen in tube yang you imagine reaction dalam tube DCCTP tadi tu.
Replication continues. However, when a dideoxyribonucleotide.
Okay, so you imagine ada banyak molecules of template ni, ini satu molecules, okay?
It is inserted, strand synthesis is terminated. The contents of the reaction tubes are then transferred to 4 lanes of an electrophoresis gel, and the oligonucleotides are separated by size and.
Okay, so bila dia DDCTP being added, dia tak boleh continue synthesize lah. Okay, dia akan stop.
Nucleotide type. The shortest oligonucleotide moves furthest down the gel.
Okay, so bila dah habis eksperimen tu, towards the end kita run gel, okay? And you should remember konsep gel tadi, the smallest size akan migrate paling bawah, okay?
Reading from bottom to top, 1 base at a time, provides the correct DNA sequence.
Okay, so masa you run gel tu, you ada 4 wells. Okay, so well nombor 2 ni you run tube yang ada DDCTP tadi tu lah. Okay, so sekarang you dapat result based on you baca paling bawah. So this is the sequence of DNA sequence. Okay, any question? Okay, so I will explain the process ni. Okay, so kita cakap tadi the starting material is a preparation of identical single-stranded DNA molecules. Okay, so these are the molecules yang we want to sequence. Okay, and then to the mixture kita ada 4 tubes. Okay, so setiap tube tu kita letak DNA molecule, single-strand DNA molecules. Okay, then setiap tube tu kita letak primers. Okay, so because we require the primer because to synthesize a new DNA strand that is complementary to the template. Okay, so then every tube tu kita letak 4 DNTPs. Okay, kita letak DNA polymerase. Okay, because DNA polymerase ni akan add DNTPs ni. Okay, okay, semua 4 tube tu ada all these things. Okay, so this does not occur in chain termination sequencing because as well as the 4-deoxynucleotide, okay, so each 4-dideoxynucleotide akan be added. Okay, so polymerase ni, DNA polymerase ni tak dapat bezakan between deoxy and dideoxy. Okay, so bila dia nak start synthesis of DNA strand tu, dia akan incorporate dideoxy, dia akan incorporate also deoxy. Okay, so but we know once dideoxy is being incorporated to the growing then newly synthesized DNA strand, okay, dia akan stop the process of DNA synthesis. Okay, okay, so this is to show you. Okay, so this is what we need to do. Okay, so you ada sequence template strand DNA yang you want to sequence. Okay, so you imagine this is tube yang kita label sebagai DDATP. This is what happened. Okay, so you add primer. Okay, and then okay, dia ada banyak molecules dalam tube tu. Okay, so for example, molecule number 1, okay, dia punya DDATP being added dekat T yang number 2. Okay, so dia akan stop the synthesis. Okay, but molecule number 2 pula, DDA ni akan stop dekat molecule T number 1. Okay, and so on. So this dia akan produce different length of DNA fragments. Okay, so this is what kita akan separate tu, kita akan separatekan pada gel. Okay, so the mixer is then loaded into a well of polyacrylamide gel or into a tube of capillary gel system. Okay, so dia akan separate based on size. Okay, so this is a vector diagram. Okay, to show you. Okay, to separate, nanti kita akan nampak lah. Dan you boleh baca sequence, okay, of the yang kita nak sequence. Okay, boleh? Ataupun sekarang ni kita pakai sequencer, okay, dia akan detect dengan fluoresce. Okay, sekarang dideoxy sequencing ni can be automated lah. So bila you hantar sequencing, dia akan keluar chromatogram macam ni. Okay, untuk nucleotide number G, dia akan keluar color pink warna hitam, T warna merah, C warna biru, and A ni. So this is the result of sequencing. Okay, any question?
Doktor, primer tu kita dapat dari mana?
Primer tu kita add, kita synthesize. Sebenarnya kita boleh synthesize primer ni, macam kita kata kita design the primer, lepas tu company boleh synthesize. Dia akan buat sequence ada A, T, T for example.
Okay, faham.
Alright, okay. Alright, so this is just to show you some of the organism yang kita dah sequence lah. Okay, with the different size lah. For example, bacterium, of course, the size of the genome is smaller. Okay, for example, this mycoplasma, 0.58 megabases je. Okay, compared to plant, okay, with 16,000 megabases. Okay, so for example, sekarang dah banyak organism yang kita dah sequence lah. Okay, alright. So to understand lagi ni, sekarang kita ada banyak next generation sequencing technology. Okay, so I will attach this paper for you to read. Okay, because for you to understand better lah. Okay, but the concept is actually in my slides lah. Okay, because you can be exposed to all this technology and the latest technology lah. Okay, so like I said, teknologi sekarang ni sebab with the advancement of technology, sequencing ni dah jadi cepat and also cheaper. Okay, and also sekarang kita ada what we call as RNA sequencing, kita sequence the RNA. Ada tools yang kita selalu guna juga, microarray, okay, and also real-time PCR. Okay, to mention few tools lah. Okay, so any question? Any question? Tak ada? Okay? Semua faham? Boleh like I said, alright. Lagi satu saya ada soalan yang macam tak straight to the point lah. Okay, so the key of your exam ni nanti, I hope you read the question carefully. Okay, saya pesan carefully. Okay, what I want to, you have to read carefully. Baca instruction carefully. Okay, but I believe 1 jam, 2 jam setengah tu should be enough time for you to answer. Okay? Alright, my paper, the first paper ke bukan? First paper you all, paper apa? Herbs. Okay. Herbs senang je. Okay, so tak ada question. Alright, apa kata kita start pukul 10? You take a break dulu. Sebab saya rasa berat nanti you tak boleh nak follow saya punya ni. Okay, kita start pukul 10 balik, boleh?
Boleh, Doktor.
Alright, okay.
Doktor.
Alright, kita jumpa pukul 10. Saya bagi link lain
Alright.
Saya register dulu